All right, everyone, let's get started. Welcome back to the afternoon session, the last session of today's event. We have three very exciting talks today, and it's my pleasure to moderate the afternoon session. Our first speaker is Neal Rosen. Dr. Rosen is a member in the Department of Medicine and in the Molecular Pharmacology and Chemistry program at Memorial Sloan Kettering Cancer Center, where he also serves as head of developmental therapeutics. He is also professor of pharmacology, cell biology, and medicine at Cornell University Medical School. His major interests include the study of key molecular events and growth signaling pathways responsible for cancer development and the use of this information for developing mechanism-based therapeutic strategies. He played a very important role in the development of tyrosine kinase inhibitors, among many other contributions and discoveries. And he received his undergraduate degree in chemistry from Columbia and an MD and a PhD from Albert Einstein College of Medicine. So it's really a pleasure to welcome Dr. Rosen here today, and we look forward to your talk. Thank you.
Thank you. He was supposed to just say Neal Rosen from Brooklyn, New York, but he did not do that because he's a normal human being. It's a pleasure to be here today. I see we're past slide one, so let's go wherever. Let's begin with I was born. It's a pleasure to be here. I thank the organizers for inviting me, and I don't really know about resistance, but that was the title of the symposium, so I decided to look at my data and see if I knew anything about resistance. I'm going to talk today about primary resistance to RAS inhibitors. Don't take the title all that seriously, but I think we got some new ideas, and we tend not to study primary resistance. Primary resistance is the targeted drug just doesn't work, and we're not going to think about why. But thinking about why turns out maybe to be interesting. I also want to tell you before I do this, surprisingly, this is just about all unpublished, so you won't find a place to look it up. So if you have a question or want to see the data again at your leisure, give me an email and you'll get it. So pancreas used to be almost as bad as glioblastoma to treat, but then God created K-von-Chokat, and we got RAS inhibitors, and we're still stuck. But so there are three kinds of RAS inhibitors in clinic now. The first type was developed by K-von's company, binds to the GDP inactive form of RAS, and the way it works, this is the only published part of it we figured out. The way it works is by trapping the enzyme in the GTP-bound state by covalently binding to the SAS domain in RAS. I don't want to go through this at all. I'm not going to talk about this today, except that everybody seems to have ignored that point in thinking about why SAS inhibitors work in a combination. The second type was made by a company called Revolution Medicines, which many of you may have heard of now, which developed a trimer that works, or works as a trimer. They basically took cytophyllin A and bound it to a structure that binds to RAS GTP. When this goes in the cell, it binds to GTP-bound RAS and gives you that complex. And most of this is known already, but I'll tell you, the clinical stuff, both types of inhibitors work in patients. They work in a way that is disappointing to some. But to me, we've been waiting for RAS inhibitors for a long time. The fact they work, and it's been around such a short time, is hopeful that we can do better. Having said that, this is a mature study with the G12D bound inhibitor, which are now called off inhibitors. And you can see that there's a considerable portion of patients don't have much happen or they progress while they're on it. Another, maybe third, have stable disease or retardation. And another third, or maybe 40%, have what they call resist responses. And these last a median of about a year, about 6 months, sorry. So the question is, why can't we do better? And what I'm going to talk today is just what I do, cloaked in the guise of resistance, primary resistance. The question is why these don't respond and why these come back. These come back for the most part, as far as we know, due to a plethora of mutants that prevent the drug from working, and these are very obvious as to what they are. Other RAS mutants, RAS alleles that revert to another RAS mutant that this doesn't bind. The G12C inhibitors are extremely selective. The second type of drug we have now, a couple of them are fairly selective and covalent, but most of them are not that selective. And the third kind, which had everybody scratching their heads and sort of being confused, is the third kind is a pan-RAS inhibitor. All RASes, all mutant RASes, NRAS and HRAS as well. And they are not particularly toxic. And they can be used anywhere. And in pancreas, in early trials, they are working in a variety of genotypes, mostly G12D. But again, the responses are temporary, which is not a big surprise if you've been doing this for a while. So what we have, the drugs always inhibit ERK. And in our models, they always inhibit ERK quite efficiently. They don't. Feedback reactivation of ERK, which occurs with MEK inhibitors and with the G12D inhibitors, is much less prominent with the PAD inhibitors or with the ON inhibitors, and I'll show you that in a minute. We wanted to study basically how to do combination therapy that would work by increasing cell death. That's the way we started this. And we just, actually, the secret, dirty little secret is we like using these drugs to figure out what RAS is doing in the tumor, what its function is, and what the results will be of inhibiting those functions. So here are two different off drugs, and you can see what you see in, and this is all cell culture. When we get to animals, I'll tell you. What you see is something that mirrors what you see in patients. You get a variety of, there's a diverse set of responses around the mean. Some are very sensitive, some are much less sensitive. If you look at what might be responsible for the diversity, we found something that was heartening to me because we were thinking about this about 10 years ago. We find, first of all, that ERK really, inhibition of phospho-ERK is really not significantly related. And this is sort of, it doesn't have the connotation you might think. They always inhibit ERK, and they always inhibit 95% of the ERK, so the small differences do not come out statistically in correlation. They might mean something. What means something is what is telling is the ability to inhibit AKT does have a high R value, and the ability to hit downstream targets of mTOR, which is obviously downstream of AKT, are quite significant. And this was heartening. I'll talk about this in a minute. I had to admit this. The paper we wrote 10 years ago in Cancer Cell, which showed that in order to work, less sophisticated drugs in combination required inhibition of both pathways. And even worse or better, and I didn't emphasize this in the paper because I thought it was crazy. If you, for those of you who don't know, PI3K activates AKT, activates mTOR. mTOR phosphorylates lots of stuff, but in terms of growth, two of the important things are S6 kinase and 4-EBP, which controls cap-dependent translation. So to our astonishment, and we'll get back to this a little later, when we knocked down 4-EBP1, which we were inhibiting in the combinations that worked, the cancer cell responded to nothing, or nothing targeted. Inhibiting RAS genetically, inhibiting RAF and PI3K with drugs or in combination did not work. The cell did not care, suggesting that the inhibition, the activation and inhibition of cap-dependent translation is a key feature of the transformed cell that must be inhibited to get stuff to work. And I would take that for what it is, and you can look at the paper, and maybe it's 10 years old and you can get around some of those conclusions, but I think it'll be hard to get rid of them completely. You don't have to look at the West. I'm constantly pilloried for my use of Western blots, so I will try not to do it too much. The ON inhibitor, on the other hand, works much faster because when it binds, it immediately disrupts the interaction of the activated RAS with effectors. It works faster, it's more potent, and it's much more potent against the RAS cells than the G12D inhibitor is, but they're still diverse, and the diversity follows the same order as it does with G12D. The most sensitive ones here are the most sensitive ones there, the most insensitive ones are the most insensitive. And this is the case even though, and this is a slide that I liked because PERC is not a good quantitative measure of pathway output. So if you look, there's 3 bands here and 3 here. So you can see the feedback reactivation you get with the G12D inhibitor. You really blunt that, but do not abolish it with the PAN inhibitor. Even so, it's not that much better in a dish than the G12D inhibitor. If you look at what correlates, once again, S6K and 4-EBP inhibition correlate. If you say maybe that means something and you add a variety of different PI3 kinase pathway inhibitors to the RAS inhibitor, you find that all of them, whether they are TOR inhibitors, PI3K inhibitors, or AKT inhibitors, they all synergize. And this is in the setting of a lung. Pancreas is a little different, and hopefully I'll get to that. But what works best is an inhibitor we made in collaboration with Revolution Medicines and Kevon, an inhibitor that inhibits TORC1 kinase selectively. And we made that not for potency reasons, not for anti-tumor activity, but because TORC2 inhibition is responsible for the hyperglycemia you see with pan-TORC. So this drug worked in that way. We succeeded in that. Those inhibitors are very potent. They have the same anti-tumor activity by themselves as pan inhibitors, and they don't cause hyperglycemia in mice or in our model system for mice, human beings. So we thought maybe we could add that to a selective RAS inhibitor and get stuff to work. So this is the stuff that impressed me, even though it's mice. These are all PDXs, or some of them are cell line. And what we see is in 7 out of 8 of these models, we've now done it in about 12 models for a variety of reasons. Lung cancers with KRAS G12C the combination is synergistic. It gives you complete or near complete remission, and we haven't seen resistance develop in that case. So even in cells where the RAS inhibitor by itself works very well, you get relapse at 40 days, but not if you have the other drug around. And in some tumors, the RAS inhibitor neither inhibitor works very well, but you get remarkable. Well, for me, I've been doing this a long time. Anything that works is remarkable, so excuse the adjective. It works very well. The only one where you don't get this regression and stable no growth is one that post hoc we looked at the genotype and found it also had an activating mutation of MEK2, which maybe explains why it doesn't respond as well. And it's not toxic, at least they continue to maintain weight. So why is this happening? And this is a 10-year-old paper. We did a lot of experiments on this, and we basically reasoned that there are a group of downstream processes that are regulated by both pathways. And that maybe, if you want to speculate, accounts for robustness of the pathway and of the cell when you inhibit one. When you inhibit one pathway, you activate the other. That is true in tumors, and it's true in humans. So what are these processes? We basically identified 3 then that we could identify. I'm sure there's 38, but translational initiation, regulation of apoptosis, and D-cyclin expression. And I'll show you a little bit more about this, but basically what happens is, I'll show this particularly, with the RAS inhibitor, with inhibiting MEK, you induce BIM. With inhibiting TOR, you inhibit inhibitors of apoptosis like MCL1. If you inhibit both, it works. If you don't, it doesn't work. This is just a neat little exception, and not an exception, but it's powerful recognition. Here's another way. Sometimes one target in the pathway is regulated by both pathways. So BAD is phosphorylated by ERK at serine 112, and by AKT at serine 136. You have to hit both to release BAD from 1433 and cause death. If you make the double alanine and put that BAD in, cells just completely die. If you make the single mutations, you become sensitive only to the opposite pathway. So I'll do this real fast because I don't want to overstay my welcome here. So it turns out that the combination of, I have to read it myself, combination of KRAS and TOR produce a higher percentage of G1 arrested cells, a bigger reduction in S phase, and much of this base is based on inhibition of cyclin D expression. Now, I will tell you in a minute, and I will pass the slide very quickly, that the TOR inhibitor actually increases cyclin D1 RNA. I just want everybody relying completely on RNA-seq to realize that that is a common phenomenon. And the MEK inhibition or the RAS inhibition gets rid of the message. But the PI3K inhibition inhibits translation. So that's good. And I'm going to say a lot about this later. But I'll say now that we think with modest evidence that 2 of the key targets of RAS activating and inhibiting and targets of its inhibition that have to be inhibited are G1 progression, and that's mediated mostly by cyclin D1 and some and cyclin D3, and a lot by things I don't know about, but that's what I believe it's true, and also the cap-dependent translation. So let me get rid of that. Secondly, the combination significantly induces death, and as I said, you induce BIM with the RAS inhibitor, you get rid of MCL1 with the RAF inhibitor. Wrong, I left that out completely because it's easy to say. And if you knock down BIM, you don't need the RAS inhibitor. And if you overexpress MCL1, the whole thing doesn't work. Genetically, with manipulations, it makes sense. The last thing, which again, I told you before, and there's a massive data on this, which is available to anybody who's interested in this stuff. You inhibit cap-dependent translation, and you inhibit the, where is this? You inhibit formation of the cap very quickly with the combination of both drugs together, is on the right here. And so you inhibit assembly, as I said, and sort of cute for anybody who works on this. When you do this, it turns out that EIF4G, which is a component of the initiation complex, I didn't know this when I started, has a couple of caspase 3 sites. So when you induce apoptosis, you get a feed forward and you cleave in a ZVAD-dependent way, a ZVAD-sensitive way, you get rid of EIF4G. So that competes for the phenotype. So this is where we are. We have a combination that does very well in mice. And if any of you have any mice with lung cancer and G12C, bring them over. At the same time, we found that this is very much work in progress for the last 3 to 5 months. At the same time, we find that these are mostly true in lung cancer and pancreas cancer with RAS mutation and a variety of RAS alleles. And I'm sure there's differences between the alleles, but this is work in progress. The interesting thing is, or interesting to me, is we find 3 classes of tumor, of the 3 solid tumors. We can organize them in 3 ways. One is our tumor models, all culture except a few animals now, where this is corroborated. In one class, you inhibit D very well with just the RAS inhibitor. And in those, you also inhibit 4-ABP. And I don't want to say you don't need the other drug, but in animals, you get small effects of the other drug. Okay? So it's as if this was working, and it's really what we saw at the beginning. The ability of it to work is dependent on its driving PI3K as well. What accounts for the second type, we don't know. Well, actually, so here's the second type. I should tell you what that is. In the second type, that's just the first type, and this is a model in culture. I have some in animals where the RAS inhibitor works very well by itself, and you don't get much with a PI3K or an mTOR inhibitor. The second type, you do not inhibit RAS by itself, but when you combine it with the mTOR inhibitor in the lung and colon, and in pancreas, anybody who works on pancreas, I'm very confused about regulation of PI3K in pancreas. I'll admit that from the stage. But you basically, in these cells, you need both drugs. This is what we used initially, what we found. You need both drugs to inhibit D. And it shows you here, this is the drug by itself in the class 2s, and this is the drug with a TOR inhibitor. Whoops. If there's anybody from stand up here, this is actually the wrong title. This is a actual pancreas model, KP64 or 6 or whatever, KP4, which is resistant to everything. This is in animals. It's what I showed you before. If you give this one, mTOR works better than PI3K. The RAS inhibitor relapses at 37 days. The PI3K treated relapses about the same time, but a little less. The combined ones stay. Okay, so I would say this is the first part. But I am, exaggerating maybe the interpretation of this. I think combined with papers recently from, what's the name of that woman? His name I always forget, at Hopkins. You must know. A very fantastic pathologist at Hopkins has shown that we all have premalignant lesions of pancreas. Laura Woods. I'm sorry, I'm old. We have lots of these premalignant lesions contain RAS mutations. Some of them contain p53 mutations, and they are not cancer. They are not growing. They are not invading. Maybe they will turn into cancer next Tuesday, but they're not cancer. And we have ignored this. I know that GEMS, you give the model, they go bye-bye, but you give RAS, but that's not the way it works. So my idea here, which is very nascent, is that there are other things that happen even before the tumor that are required. And some of those may activate D better, so you grow faster. Some activate ERK better. Tyler has a paper on colon CA, a colon gem, where with time you increase ERK, and that's when you get the tumor. What's the other piece of data? And David Tubeson has tissue from patients during progression. ERK goes up. KRAS does not go up, but ERK goes up. Nobody knows why. So we are busy now trying to figure out why the RAS is less sensitive to cyclin D there and why two are required. What's occurred? Is that are those events that create complexity and robustness or allow transformation or not? I don't know. Last thing I will tell you is, do I have any time? This gives me
Understand.
Oh, wow, 20 seconds. I can do the Gettysburg Address. This is my first opportunity to show an acknowledgement to my son, who's an assistant professor at Sloan. So the idea here is that drivers do something, and most drivers deregulate G1, and they do it by D. And I'm postulating, without more data than I've shown you here, that most drivers activate, or some driver has to activate PI3K-mTOR. So we did something else, and it's going to be hard to explain why, because I don't remember why, but we were studying regulation of estrogen receptor translation in breast cancer and anywhere, and we found that it is regulated, it is not TOR dependent, which I think makes you resistant to the effects of starvation during pregnancy, or somewhat resistant. So we took an EIF4A is required for translation. So we gave it's a helicase that is required for complex 5' UTRs, and we gave this drug, and we found that it's synergistic. I'm available later to ask anybody the whole story, but it does in animals inhibit ER. I had a gigantic idea that any school child could have. Fulvestrant induces ER degradation, so together they should synergize, and they do. And then we started a clinical trial driven by Ezra Rosen, who I think I'm related to. And this is what happens, basically. This is not everybody. This is just the pitiful amount of biopsies we have. But this is what happens. Circulating DNA goes down, and pre-treatment ER goes down, and it looks interesting. The second thing, for those of you who know, this is the obligatory clinical slide of something that happened once, but this is not something that happened once. There's a woman, a surgeon actually at Sloan, who's seen at Sloan, who had tumor all over the place, and this is what it was like a month later. Okay, so that gets my attention. And so we, for reasons known only to my son, this is the most exciting part of this. He added a CDK4 inhibitor, which makes molecular sense, but it's medically nuts. Because I should say that it's bad enough, or you're lucky enough, that the EIF4A inhibitor is not particularly toxic, but adding the CDK4 inhibitor does not add any toxicity. I'll take suggestions on this. I have some ridiculous ideas. But with the two drugs, you get about a 30% disease control rate. If you add the CDK4 without adding tox, you get a 74% rate. And the idea here is, of course, we're only inhibiting one enzyme. We're inhibiting cyclin D CDK4. We're inhibiting two components of the complex. And that's what's going on. But now here's for the grandiose part. Oh, and in culture, and I should say EIF4A also regulates CDK4 and CDK6 synthesis. And we think what's going on in terms of lack of toxicity, it's the only thought we have, is these are all very rapid half-life proteins. And those are the ones you're most likely to hit at an intermediate level of the drug. So here is my, what I get out of this. Well, you ought to be able to, if you can replace the fulvestrant, which is not, it's toxic, it has toxicity to women, but they're not like cancer drug toxicity. If you can replace it with oncogene, with inhibitors of the oncogene, that drug for which it's very important to hit cyclin D CDK4, i.e., anything with RAS or RAFs, and probably a lot of other things, you could do the same thing. We are actually, for all of you who do this kind of thing, we're in a very peculiar, almost unknown position here. We have something that might inhibit the tumor, but we know it's not toxic. We never know it's not toxic. So that's what we're doing. And the scientific thing we're doing is trying to understand, in terms of pancreas and colon transition, evolution, the mechanisms whereby regulation of cyclin D1 and D3 changes over time with development. So that's the last thing I'm going to say. I'm only a couple of seconds over, thank God. This work was done by a great postdoc, Philip Choi, and a great grad student who now works with Josh Rabinowitz, Jacob Boyer. The clinical was driven by Ezra Rosen and Nicholas Mice. The best part is a collaboration with Sarat Chandalapati. All of it is a collaboration with Elisa Di Stancina. And the companies, I mentioned Revolution Med for the TORC drug. I did not mention, and my fault, Effector Pharmaceuticals that developed the EIF4A inhibitor. Thanks very much.
Okay, thank you. I think we have time for one question. Then should probably move.
Nobody wants to ask anything. Come on, destroy me.
Of course not. So I'm going to ask the clinical translational question that will almost certainly come up is patients get resistant to the RAS inhibitors, right? Because everyone's giving RAS inhibitors for certain diseases. And then when they fail, temptation from your talk might be to give rapamycin because we can just write that off label right now. Do you think that's a good idea or not a good idea?
Okay, first of all, animal studies with rapamycin are worthless because it inhibits the angiogenesis when you inject subcutaneous tumors. So it gives you the wrong answer 100% of the time. Secondly, we don't get mTOR by itself to work. mTOR works. You could say, look, I know that mutants are going to develop.
Yeah. I'm saying adding it to the RAS inhibitor.
Downstream by this, right? Well, I don't know that. One way of dealing with the mutants that occurs in 6 months is to take the Vladimir Putin and kill all the cancer cells. So that's what we're trying to do. We're trying to enhance apoptosis, ferroptosis, and every other apoptosis before those mutations survive.
Yeah.
Anybody else? Oh, well.
Well, thank you very much for your talk.
Okay, good. What's that, Matt?
Okay, our next speaker is Michael Yaffe, who's the David Koch Professor of Science at MIT. and also the director of the MIT Center for Precision Cancer Medicine at the Koch Institute. He's also a senior associate member of the Broad Institute and an attending surgeon in the divisions of surgical oncology and acute care surgery at Beth Israel Medical Center at Harvard Medical School. His work has led to the discovery of new signaling pathways and molecular targets that control tumor cell responses to DNA damage and cell cycle progression, including BRCT, FHA, 1433, Polobox, and PH domains, the discovery of both TAS and the molecular chaperone, CCT, and the creation of novel anti-cancer combination treatments based on tumor-specific vulnerabilities and time-staggered drug administration. And assuming the presentation comes
There we go.
up, I'll hand it over to Mike. We look forward to your talk.
Thank you, Anders. I know it's traditional to thank the organizers. Doesn't seem to be much point in my doing that. What I want to tell you about today is some systems pharmacology and CRISPR-based approaches to trying to understand heterogeneity and drug resistance in cancer treatment. And a lot of what we work on in our lab is really focused on a few tumor types. Ovarian cancer is one of the tumors that we really care a lot about, that and colorectal cancer and appendiceal cancer. And in particular, what we've been interested in is trying to understand how DNA damage and protein kinase signaling pathways work together to control tumor sensitivity or tumor resistance. I'm going to try to make 3 key points today, all related to resistance and heterogeneity, but although we heard a lot about genetic heterogeneity, I want to talk about heterogeneities of other types. So the first point I want to make to you today is that current combination treatments that we use in cancer are not synergistic, and they're not even additive. They definitely work, but they work for a different reason, and that's heterogeneity at the organism level. And then I'll show you how the lack of tumor biomarkers means that we're subjecting many patients to toxic and adverse effects without clear benefit. Okay. I'm going to start with a combination therapy that we use all the time in colon cancer and pancreatic cancer. If you know someone who is diagnosed today with colon cancer or pancreatic cancer, they are almost certainly going to get combination chemotherapy first. And that involves a cytotoxic drug that will damage DNA. And we think these work because tumor cells are rapidly proliferating, and most tumor cells have some type of defect in their ability to repair damaged DNA. And one of the 2 classic cocktails is called Folfox, and it's basically the 2 active ingredients are really 5-fluorouracil and oxaliplatin. And we know that this drug works because 5-fluorouracil, a drug that's been studied for 50 years, is an inhibitor of the enzyme thymidylate synthase. And you need thymidylate synthase to make TTP. And when you then can't make deoxynucleotides and you damage DNA, for example, by using oxaliplatin, a cross-linker, the combination of not being able to have nucleotides to do the repair in the setting of damage causes toxicity. The other possibility that is widely used besides Folfox is Folfiri, and that's again 5-fluorouracil because of its inhibition of thymidylate synthase together with irinotecan, a topoisomerase 1 inhibitor. This causes DNA double-strand breaks, which in the absence of an adequate amount of nucleotides cannot be repaired. And so there's a logical basis for combining 5-fluorouracil with oxaliplatin or irinotecan. When Carl Merrick was a postdoctoral fellow in the lab, we were very interested in trying to understand how we could make that combination therapy even more effective. And he made a discovery that I found very upsetting, and that was if he looked at cell viability as a function of 5-fluorouracil dose, you got the red curve. And if he added oxaliplatin, just like we do in the clinic, he got the blue curve. The problem is that the curve he should have gotten is the gray curve, because the gray curve is what you get if you just add these 2 drugs together. I'm going to show you some heat maps where I'm going to show you increasing concentrations of oxaliplatin this way, increasing concentrations of 5-FU this way. And in these curves, we're going to look at the difference in cell death between what you would expect and what you would observe. The more the blue color means the drugs are more antagonistic. They're not synergistic. They're not additive. If they were additive, everything would be white. And I think you can see here that in 8 different colon cancer cell lines, all except for 1, gave you less than additivity from combining these drugs. One drug was antagonizing the ability of the other drug to kill cancer cells. Maybe this is an artifact of cell culture, except it's not, because we teamed up here with Adam Palmer at UNC to look at real clinical trial data. And what I'm showing you here is the survival, progression-free survival of patients that receive with colorectal cancer that get oxaliplatin alone, that's in blue, or 5-FU and leucovorin alone, that's in brown, or the combination, and that's in red. And what you would expect if you build a synthetic cohort and you simply populate it with patients and ask, what would I expect the survival to be if I simply added these drugs together? You get the black curve. The red curve doesn't do quite as well as the black curve. It's even worse if you go to irinotecan. Survival, progression, time to progression, on patients receiving irinotecan is in blue, in patients receiving 5-FU and leucovorin is in red. What you would expect to see is in black, and what you observe is in red. That suggests that the very drugs that we are treating patients with are antagonizing each other, and we are getting, at the level of an individual patient, less of a response than you would think if we just added the effects of the drugs together. Now, why is that? The reason is heterogeneity. Not heterogeneity at the level of the tumor, heterogeneity at the level of the patient. Some patients in the cohort are responding to 5-FU. Some patients are responding to oxaliplatin. Some, perhaps, actually do respond additively. Some don't respond to either drug. And because we don't have biomarkers for who responds to what drug, we're giving everybody the cocktail in order to be able to get benefit out of only a subset of these patients. Now, this means that, as I said, this lack of biomarkers is one of the reasons why combination chemotherapies appear to work at the population level, but don't work at the level of an individual tumor. Okay. The second point I want to make is that many effective drugs in colon cancer and pancreatic cancer actually work through a mechanism of action that is very different than what the textbooks tell us. I'm going to show you that they work by targeting RNA, targeting ribosomes and RNA translation. And Neal already commented on roles of chemotherapy in RNA translation. Well, we were very puzzled by the fact that the combination of 5-FU and DNA damaging agents didn't seem to be synergistic. And so Carl looked in patients, he looked at patients who were either very sensitive to 5-FU, to FOLFOX, or very resistant, and he ranked genes by where they scored, and thymidylate synthase did not appear to confer either resistance or sensitivity. If you looked at the trials from patients that received FOLFIRI or FOLFOX, and you coded them for those who had a response to the therapy and those who didn't, and then you asked, where does thymidylate synthase expression lie? In fact, doesn't correlate at all. There is no correlation between the known target of this drug and the response that we see in patients. Well, if thymidylate synthase is not important here, what else could 5-FU be doing? Well, in addition to inhibiting thymidylate synthase, 5-FU itself will become metabolized into either 5-fluorodeoxy-UTP, which could get incorporated into DNA and cause DNA damage, or it will become 5-fluoro-UTP, which will be incorporated into RNA, and perhaps there's an RNA damage response. Now, Carl was very clever, and he came up with a very clever way to figure this out. He reasoned that he could, instead of treating cells with 5-FU, let's treat them with either the RNA-damaging metabolite 5-FUR or the DNA-damaging metabolite 5-FDUR. And I'm going to use these colors, green and blue, to indicate what we're going to do. So everything you see in green refers to the RNA-damaging metabolite, and everything in blue will refer to the DNA-damaging metabolite. Okay. So we can compare our cells more sensitive to the RNA-damaging metabolite, that's in green, or the DNA-damaging metabolite in blue, and look at the IC50s, and I hope you'll notice that in 9 different colorectal cancer cell lines, and so everything below the line, 0 means they were equally sensitive. This is the log of the ratio of the IC50s. Anything below the line means they're more sensitive to the RNA-damaging metabolite. Anything above this 0 line means they're more sensitive to the DNA-damaging metabolite. And I hope you can see here that all these colon cancer cells are sensitive to the RNA-damaging metabolite of 5-FU, not the DNA-damaging metabolite. Now, maybe this is an artifact of cells in culture, so we teamed up with Omer Yilmaz, and George Eng, a postdoctoral fellow in his lab, was able to grow for us primary colon cancer organoids from real patients. And I'm showing you here those primary organoids for their sensitivity to either the DNA damaging metabolite in blue or the RNA damaging metabolite in green. And all the patient tumors are more sensitive, the organoids are more sensitive to the RNA damaging metabolite. Now, we can get a hint of what's going on if we look at the main location of RNA synthesis in the cell, which is, of course, the nucleolus. And if we treat cells with either 5-FU or the RNA damaging metabolite, you can see that the nucleolus becomes partially shattered, but not if we use the DNA damaging metabolite, suggesting that maybe the target has something to do with ribosome biogenesis. Now, I've shown you this already. I've shown you that in colon cancer cells, these are HCT116 cells, they're more sensitive, as shown in green, to the RNA damaging metabolite than to the DNA damaging metabolite. And if we inhibit RNA biosynthesis using an RNA polymerase inhibitor, we specifically reduce the ability of the RNA damaging metabolite to kill them, but not the DNA damaging metabolite. Carl then went on, together with our colleagues at Genentech, to do proteomics and try and understand what was happening to these cells when they saw the RNA damaging metabolite or the DNA damaging metabolite. And so we prepared lysates of these cells and we enriched them for either the phosphopeptidome or the total proteome or the ubiquitinol. Okay? And what I'm showing you, I'm going to ask you first to focus on what's in this blue box. These are the phosphopeptides. This is the phosphoproteome. If you stimulate the cells with a DNA damaging metabolite, that's the light blue and dark blue, you get a lot of phosphoproteomic changes. You don't see much happen when you use in green the RNA damaging metabolite. Now, if we look at what gets ubiquitinated, when you induce DNA damage with the DNA damaging metabolite, not much happens. But the RNA damaging metabolite results in a profound amount of ubiquitination. What are the proteins that are being ubiquitinated? It is basically every protein that forms the ribosome. Somehow, the RNA damaging metabolite of 5-FU is leading to the rapid degradation of ribosomes. And you can see that if you look at the distribution of ribosomes in the cell. I'm showing you on control conditions in black that you see monosomes, the ADS monosomes, some free 40S and 60S subunits, and polysomes. If you damage the cell with the DNA damaging drug, that's the blue curve, you lose the production of polysomes, but you gain both the ADS monosomes and the ribosomal subunits. In contrast, if you treat cells with the RNA damaging metabolite, you end up with the green curves. You lose polysomes, but you lose monosomes and 40S and 60S subunits. And I won't show you this, but the damaged RNA is being degraded in lysosomes, and the ribosomal proteins are being degraded by the proteasome. Now, interestingly, I don't want to leave you with the idea that the textbook mechanism isn't true about thymidylate synthase. It is true. It's just true in the wrong cells. And I say that because if we look at the ratio of IC50s for the RNA damaging metabolite or the DNA damaging metabolite, you can see that for some cancer cell types, far and away, the effect of 5-FU is being mediated by the RNA damaging metabolite. In other tumor cell types, it's actually the DNA damaging metabolite that's important. But interestingly, the cancer cell types where the RNA damage seems to be the dominant mechanism are the exact tumor types where we use 5-FU in the clinic, or we used to use 5-FU, breast, colon, and ovarian cancer. Okay. We can take the cell lines that you see here from the NCI 60 and rank them for those that are more sensitive to the RNA damaging metabolite or those that are more sensitive to the DNA damaging metabolite. And if we do that, we find that the cells that are more sensitive to the RNA damaging metabolite correspond to a gene set that involves RNA polymerase I promoter opening, consistent with the idea that, in fact, they're more sensitive to 5-FU because they're incorporating this into RNAs driven by RNA pol-1, ribosomal RNAs. In contrast, if we look at those cells that are resistant to 5-FU and are those cell lines and those genes that seem to predict sensitivity to the DNA damaging metabolite, those genes are the ones that are elevated in patients who are resistant to being treated with our conventional combination chemotherapy. Okay. Now, we can play around, we can manipulate RNA ribosomal biogenesis and ribosomal RNA synthesis. We're going to stain our cells with EU, and we're going to look in the nucleus because the main sites of EU incorporation in the nucleus will be the nucleolus. And if we starve our cells for glucose, you see we reduce the amount of nucleolar staining for new ribosomal RNA synthesis. And when we do that, that's shown in the black curve, we make our cells resistant to 5-FUR. We make them resistant to the RNA damaging metabolite. In contrast, we can inhibit the lysine demethylase KDM2A, and that increases ribosome biogenesis. And when we do that, we sensitize our cells now to being treated with the RNA damaging metabolite. Why do I think this is up? This is not simply true using these. We can do the same thing. We can knock down KDM2A. We sensitize our cells to 5-FU, the parent compound. We sensitize them to the RNA damaging metabolite, but we don't sensitize them to the DNA damaging metabolite. Why is this important in a context of resistance? Because when we treat cancer cells with 5-FU, we're preferentially incorporating it in the tumor cells that respond to 5-FU into ribosomal RNA, leading to degradation of ribosomal proteins and degradation of ribosomal RNA and RNA-induced cell death. The reason I think this matters is because when we give patients oxaliplatin and irinotecan to induce DNA damage, we know that one of the things that does is suppress ribosome biogenesis. We are inducing drug resistance in cancer ourselves by simply using the wrong combination at the individual tumor level. The right combination at the population level, not at the individual tumor level. So I've shown you that many effective drugs work by targeting ribosomes, RNA, and translation, which is, I think, a relatively unexplored therapeutic space. But really, my passion these days is for ovarian cancer. And I want to show you some new data that RNA damage and translational regulation actually control sensitivity and resistance in high-grade serous ovarian cancers. And we did this by doing what I think was a pretty clever CRISPR screen. Now, let me warn you that most CRISPR screens don't mimic conventional cancer treatments and are therefore have the potential to mislead us. I say that because if we use Cas9 in order to inactivate a variety of genes, right, in a population of cells, most of the studies I know of then expose those cells to high-dose continuous chemotherapy and then score the surviving cells for guides that are either upregulated or were lost in order to identify genes that played an important role in the process. The problem with this approach is let's think about how we treat ovarian cancer. We don't treat ovarian cancer patients for 3 days of continuous high-dose drug exposure. They actually get a high dose of cisplatin over 1 hour, a drug that has a half-life of 30 minutes, okay? And then they get, over about 3 hours, paclitaxel, a drug that has a half-life somewhere around 4 hours. And then they get nothing for 3 weeks. And so to try to mimic the drug effects, we decided to do a CRISPR-I screen. This is CRISPR inhibition. We're going to use Cas9 fused to the CRAB domain, something which is going to function as a transcriptional silencer. So instead of making genetic nulls, we're simply going to be able to reduce the expression of these genes. That lets us interrogate essential genes, but more importantly, I think it mimics drug targeting, where you're not inactivating a gene down to 0, but you're only partially inhibiting it. And so the way we did this was to use an sgRNA library that was optimized for CRISPR-I, and then treat the cells with the library, expose them to chemotherapy for only 4 hours, wash the drug away, and then let the cells grow out for 9 days, much closer to what happens in the clinic. And we did this under conditions where we got about 50% growth inhibition for control guides. And then, of course, depending on what we knock down, some of the cells will die, some of the cells will stop proliferating, and others might proliferate more. And so when we did this analysis, we did it using 2 different drugs, cisplatin and oxaliplatin. These are both platinum drugs, but for reasons that are unclear, cisplatin or carboplatin work very well in ovarian cancer. They don't work well in colon cancer or pancreatic cancer. In contrast, oxaliplatin works pretty well in colon and pancreatic. It is not a good drug to use in ovarian cancer. And what emerged, what I'm showing you here in blue, are genes whose knockdown sensitized the cells, and in red are genes whose knockdown made the cells resistant. And some of the hits were the same, and some were different. Now, a trick about CRISPR screens, you cannot believe what emerges from the screen. You have to validate it. And the way we validate this is all of our ovarian cancer cells that express dead Cas9, which is what we need in order to do the screen, contain GFP. And we put in guides, either relevant guides or irrelevant guides, in the presence of BFP. And so we can look at the ratio of GFP to BFP cells. Because if the gene we knock down sensitized them to a drug, then the blue cells will drop out, and we will see a ratio that reflects more green cells than blue cells. If the gene that we knock down makes the cells resistant, then the blue cells will overtake the green cells, and we'll see an enrichment of blue cells over green cells. Now, we're going to normalize everything here to an irrelevant guide. So I'm showing you in light orange and in light blue the vehicles, and in dark orange cisplatin, and in dark blue oxaliplatin. And here's the results that we got. Okay? So the genes that turned out to dominate sensitivity to either cisplatin, that's the orange that you see here, or oxaliplatin, that's the blue. And I'm showing you here the GFP-BFP ratio. So all the vehicles had, you can see all the vehicles normalized to 1. But you see that the genes that really seemed to dominate the response to cisplatin or oxaliplatin were genes that were part of the pathway of DNA repair through homologous recombination, or the pathway of DNA repair through the Fanconi anemia pathway. Now, why is this interesting? Well, it's interesting because if you read the literature, what most platinum drugs do is introduce intrastrand crosslinks, a crosslink within one strand. These are preferentially repaired by either nucleotide excision repair or translesion synthesis or base excision repair. Only a tiny fraction of the DNA is subjected in a platinum drug to an interstrand crosslink, a crosslink between the two strands. And it's those crosslinks, particularly during replication, that require the Fanconi pathway or the HR pathway. And so what this told us was that even though the bulk of the effect of the drug is repaired through these other pathways, those pathways were actually not important in controlling the sensitivity of these cells to the drugs we use. What really controls their sensitivity turned out to be homologous recombination and the Fanconi pathway. This is important because patients whose tumors who have ovarian cancer that's defective in homologous recombination do very well with PARP inhibitors. And what our data is saying is that we shouldn't just give them a PARP inhibitor after the therapy. We should be combining the PARP inhibitor and the platinum drug at the same time. Okay. What else came out of this? Well, particularly for oxaliplatin, many of the targets turned out to be things that are involved in RNA biogenesis or RNA processing. And I'm going to show you hits from one of those here. Okay, so to really interrogate this better, we did a second CRISPR-I focus screen, focusing here on RNA binding proteins. Again, we used only a short exposure of the drug, then followed by drug removal, and we followed this by 2 days of culture because we didn't want long-term effects as a result of manipulating RNA levels. And so 2 days later, we looked under conditions where we inhibited about 70% of the growth for control guides. What emerged from this analysis? A whole bunch of RNA binding proteins seemed to cause sensitivity to oxaliplatin, cisplatin, and paclitaxel, or caused resistance. I'm only going to focus on one here, and that's this gene ADAR, adenosine deaminase, which actually made the cell sensitive not just to platinum cross-linking agents, but also to the antimicrotubule drug paclitaxel. Okay. Now, what does ADAR do? There are 2 isoforms of ADAR. ADAR is believed to be involved generally in editing double-stranded RNAs, for example, viral RNAs. And in the absence of editing, you turn on an interferon response and you turn on a regulatory pathway that limits translation, ultimately resulting in cell death and the production of inflammatory factors. In addition, if you can edit the RNA, not only do you suppress this, but you promote the degradation of R loops. And so we then went back and now interrogated the effects of knocking down ADAR. Turns out it's a pretty essential gene. You can only knock it down to a certain extent. But here, our ADAR knockdown, I hope you'll notice the dark orange and the dark blue, every ADAR guide that we used, this shows you the level of ADAR that we were able to suppress. The more we suppressed it, the more we sensitized it, and we often sensitized it a little bit more to oxaliplatin than we did to cisplatin. Furthermore, suppressing ADAR resulted in the accumulation of R loops in those cells after they'd been treated with cisplatin. Finally, we were able to look at a data set from patients with high-grade serous ovarian cancer who were treated with platinums, drugs, and taxanes. And just like the cells where if you reduce the level of ADAR, they became sensitive, patients who had low levels of ADAR expression did pretty well as far as progression-free survival. In contrast, those patients who had high levels of ADAR were the ones who did quite poorly over time. Okay. Now, interestingly, there are a lot of roles for ADAR, and I'm hoping that you might hear about one of them from Kate, but my friend Nick Haining actually published a paper recently that showed that if you don't have ADAR, not only are we showing you that they're more sensitive to drugs, but in fact, you seem to overcome the resistance to immune checkpoint blockade. Okay. Now, I have 1 minute left, and this is a published work, so I'm going to spend 1 minute on it. I just want to remind you that the timing of drug co-administration actually matters, and that you can harness tumor cell plasticity that we heard about in the first half of the symposium by something we're going to call dynamic therapeutic rewiring. This is published, so I just want to summarize it briefly. If you take triple-negative breast cancer cells and you treat them with the EGFR inhibitor erlotinib, not much happens. If you treat them with doxorubicin, the topoisomerase inhibitor, not much happens. If you combine them, you get a trivial increase in the amount of cell death. But if you block the EGF receptor and you just wait, and 24 hours later, now you give them the topoisomerase inhibitor, you increase the cell death over 400%. Now, it's very specific to triple-negative breast cancer cells. If you do this with cells that overexpress HER2, then the time-staggered combination is actually worse than giving the topoisomerase inhibitor alone. And I'll simply summarize this because it's published to say that we teamed up with Paula Hammond's lab. We made nanoparticles that did this time-staggered release. You can see here in mice with flank tumors, if we treated them with our drug that gives the time-staggered release, you can see we got a profound decrease in the tumors. If you just give nanoparticles that release doxorubicin alone, the tumors really don't care. It turns out that this is because what erlotinib is doing is rewiring, dynamically rewiring the state of the cell from a state of the cell in which the inhibition, in which DNA damage can cause death through only one pathway, and we can now unmask a second pathway when we block erlotinib. This suppression of a caspase 8-dependent pathway can be blocked if you chronically suppress erlotinib. The sensitivity, which I'm showing you here shaded in colors, the darker color means the more sensitive that they are, does not correlate with the levels of the EGF receptor, but it does correlate quite nicely with the phosphorylation of the EGF receptor. And this gives us, well, let's go back. And this gives us both two things. It gives us now a biomarker for who to treat. Don't treat people with high levels of EGFR receptor. Treat patients who are showing activation of the signaling pathways through that receptor. And it gives us a biomarker for response. We can now look serially at whether caspase 8 has been unmasked as an indicator of whether our therapy works. In the lab right now, as I said, we're very focused on ovarian cancer. I think about it day and night. We're currently working on doing combination therapies using a PLK1 inhibitor, a therapy that has only modest effects on its own. Okay, so I've told you three key things, I hope, today. I've told you about combination chemotherapy. I've told you about this therapeutic space involving RNA and RNA editing and translation. And I've told you about the timing of drug co-administration through harnessing tumor cell plasticity and dynamically rewiring to stack those tumor cells in a particular state that is particularly responsive. I will stop here and just tell you a little bit about who did the work. Yi-Wen Kang co-manages the lab with me. I couldn't do anything without her these days. All the work on 5FU was started by Carl Merrick and done by Johnny Chen. And all the work I told you about the CRISPR screens really was championed by Yi-Wen, begun by Erica Hanley, and has been followed through by Molly Bird, Robin Liu, and Jack Schaefer. I'll stop here. Thank you very much.
Okay, I think we have time for a couple of questions. Yeah.
Great talk, Mike. I have a question about the first story that you presented. The missing transcripts in that model were Part2 transcribed RNAs.
PAL1. So all
No,
of them,
you had POL1 and POL3 RNAs as part of the model.
That's correct.
What makes POL2 transcripts not sensitive to the RNA damaging effects
So
of
it's not that
5FU?
they're not sensitive. It's that they are. They certainly become corrupted. They certainly incorporate 5FU. It's just that what we found seems to dominate the death response, seems to be more of the ribosomal RNA and tRNA damage than the mRNA damage. And I suspect it's because we have quality control programs built in to make sure that we don't make messed up ribosomes. Whereas mRNA, with its turnover, I think is just more likely to be tolerated and you'll make new mRNAs.
Thank you for your talk. So you mentioned promising discoveries using clinically oriented CRISPR screening. And you also talk about some serendipitous discoveries by considering timing into combination therapy. So my question is that, can we think of a strategy to combine the timing into a CRISPR screening? It's not a clinically oriented screening, but it's a novel screening strategy to discover and optimize combination therapy.
So you mean, so how to optimize combination chemotherapies by using
CRISPR screening, considering time into the screening pool.
We haven't thought, I think it's a good concept. We haven't thought that much about how to do that. I'm sure that it's doable. What we really need is some kind of a biomarker that tells us exactly what the right dose of the combination is. Right now, we've actually been focusing on how do we combine these combination therapies with immunotherapy. And I think that there's real potential there. I think you're going to hear a little bit more about that from Kate.
Thank you.
Okay, the last talk.
Have you looked at the formation of left-handed zRNA in your systems? Because you talk about ADAP-P150, which Nick showed, knocked that out, you got the same immunotherapeutic effect. You're talking about broken down ribosomes and potentially stress granules that contain zRNAs.
It's a great question, and you obviously noticed that ADAR contains a zDNA binding domain.
Yeah, that was discovered during
MIT is responsible does that. So I think zDNA probably plays a really important role here, but we have not explored that in detail.
Yeah, there are great tools available to do that.
But let's talk. Thank you.
Thank you, Michael. Okay, our last speaker of this session is Dr. Katherine Chiappinelli, who's an assistant professor in the George Washington University Department of Microbiology, Immunology, and Tropical Medicine, and also in the George Washington Cancer Center. She received her PhD from Washington University in St. Louis and pursued postdoctoral studies at Johns Hopkins with Dr. Stephen Balin, where she and her colleagues discovered that inhibiting DNA methylation causes an immune response in cancer through transcription of transposable elements. Her independent lab, which is DoD and NIH funded, studies the epigenetic changes in cancer and how epigenetic drugs can reverse these, specifically focusing on transposable elements and the anti-tumor immune response. Thank you, Kate. We look forward to your talk.
Thank you so much. And thank you to the organizers for inviting me here. It's really an honor to present at this symposium in this historic place for cancer research. And thank you all for staying for the last talk. I hope to convince you why transposable elements are important. Here are my disclosures. And because we haven't spent a lot of time on tumor immunology yet in this symposium, I just wanted to throw up this really nice background from Bob Schreiber, showing that as cancers develop, transformed cells are in the body here in orange, our immune system is able to eliminate them sometimes, to recognize them as foreign. But by the time that a cancer is diagnosed, it's undergone immune escape or immune evasion, where cancers downregulate MHC that presents antigens. They secrete suppressive cytokines. You can have expression of molecules called immune checkpoints that are going to suppress the host immune system. And in the context of this symposium, I'm going to speak about our work to potentially reverse resistance to immune therapy. So to take cancers from this state, which would be immune escape, back to elimination, to bring in immune cells and to destroy those cancer cells. And translationally, just like the previous talk, we work on ovarian cancer, which is a really deadly disease. And unlike some other solid tumors, has really had no change in the 5-year survival rate for decades. Relevant to this talk, high-grade serous ovarian cancer, the most common subtype, is characterized by almost 100% mutant p53 and very high expression of transposable elements. And despite these data, now from about 2 decades ago, showing that infiltrating T cells predict much better survival in ovarian cancer than patients who don't have infiltrating T cells, there has been low response rates to immune checkpoint blockade for this disease. And so our goal is to look beyond the coding genome for novel therapies for ovarian cancer. We focus on transposable elements, which make up about half of our genome. These were discovered by Barbara McClintock, and for a visualization here, transposable elements are defined as elements of DNA that can insert themselves about the genome on their own accord. And if you look at these corn cobs, you can see yellow kernels and red kernels, and then down here, there's some kind of mottled kernels here, and that is the result of a transposable element inserting itself into a gene that would code for the red or the yellow pigment. In the human genome, most of the transposable elements are no longer active. They have not been conserved through evolution. Here's a pie chart of their approximate abundance in the human genome. About 8% are LTR, or ERV elements. These are derived from exogenous viral infections. So think about similar to HIV, how these would be integrated into your genome. They can no longer make functional viral particles. They can make RNA, and some can make proteins. Then you have the line 1 elements, about 20% of our genome. These are the only functional retrotransposons in human. About 1% of them can still reverse transcribe and insert themselves in the genome. And then you have about 13% sign or allo elements. In general, these make RNAs that can form hairpins. In normal cells, these transposable elements are silenced by epigenetic modifications. And during the global epigenetic dysregulation that occurs during cancer, during transformation, they lose these modifications and are re-expressed. This is just a summary of a TCGA study, and so we're looking at different cancer types on the x-axis here, showing that compared to adjacent normal tissue, transposable elements are overexpressed. For example, in bladder cancer and in head and neck, but in some cancers they are not. So in general, you have enough regulation, but not in all cancers. And this is just showing enrichment of the specific families of TEs, so line 1 and ERV elements as well. And so broadly across cancers, we do see this upregulation of transposable elements. So why should we care about these? The first thing I would like to tell you about is that this is expressed, some of these Ts are expressed very early in cancer, and this is work done by Kathy Burns here at Dana-Farber, David Huntsman, and Yiming Xia Hopkins. And they showed that a majority of ovarian cancers expressed line 1, and this is an early event. So here's line 1 staining. This is a p53 signature that precedes stick lesions, which are the precursor to high-grade serous ovarian cancer. You can see that this p53 signature is mutant p53, so not null, but mutant, staining very strongly. And then in the stick lesions, we have high expression of line 1. And Kathy Burns and David Walt have shown here that they can detect high levels of this line 1 protein in the blood of really early stage ovarian cancers, and are currently following up on this as a biomarker for earlier diagnosis of multiple cancers. So because we know that full-blown cancers have high expression of line 1, we wanted to learn more about how this expression changes during oncogenic transformation. And so we used the model developed by Bob Weinberg here at MIT, going from fibroblasts, immortalizing them with hTERT, and then transforming them with SV40 and mutant HRAS. And we isolated DNA and RNA from every step of this model to determine how DNA methylation as well as expression of the TEs was changing. And this was work led by my PhD student, Thomas Conham. These are volcano plots looking at the different stages of the model compared to normal or early passage fibroblasts. So here, this is the cutoff for significance. And then this would be a significant increase. This would be a significant decrease on the left. And you can see that immortalization does not change these significantly, but transformation causes an increase in TE expression. And these are really more of the LTR or ERV elements that are increased. This is looking at methylation. Concomitantly, we see a loss of methylation. So hypomethylated regions, losing methylation, are much more, there are many more of these in the transformed cells than the immortalized cells. And we see very little gain of methylation, which goes along with what we know about cancer. There's global loss of the methylation and focal gains of methylation. When Thomas combined the methylation and expression data, so here on the x-axis, we're looking at a gain or loss of methylation, and then the y-axis, a gain or loss of expression. you can see that many TE families are up here in this corner, which will be loss of methylation and gain of expression, as well as if we look at individual loci, loss of methylation and gain of expression, implicating DNA methylation as controlling these regions. And when we take the TEs increased in this model and cross-reference them with those upregulated in TCGA, we see an overlap of about 9 ERVs here that are both upregulated in TCGA samples, as well as in the earliest stages of our model. And we also see similar hypomethylation looking at fallopian tube models, so a little bit more relevant to ovarian cancer. We also see loss of methylation at these regions. And so we're currently pursuing these ERVs as potential biomarkers for early diagnosis of this disease, as well as understanding more about what's causing them. The second reason I think we should care about transposable elements is their potential use in cancer immune therapy. And the reason I got into this field was working in cancer epigenetics, and the initial impetus for using epigenetic drugs was to take tumor suppressors that were turned off by different modifications, like DNA methylation or suppressive histone modification, and reversed these silencing marks to activate tumor suppressors. The drugs I will discuss here, DNMT inhibitors, abbreviated AZA and DAC, as well as HDAC inhibitors, which will inhibit the enzymes that take off this activating acetyl mark. And the overall impact of both of these inhibitors is to open up the chromatin. During my postdoc with Steve Valen, we actually showed that one of the biggest effects of DNMT inhibitors was a broad upregulation of immune signaling in tumor cells. So when you treat cells with DNMT inhibitors, a large part of what is methylated in your genome is actually these transposable elements. We think about genes being methylated, but actually a lot of what is methylated is transposable elements. So when you remove methylation at these TEs, you get transcription of transposable element RNA. This looks like viral RNA and binds to MDA5, which senses viral RNA, and this causes canonical type 1 interferon signaling, so as if the cell sees a virus, cancer cell apoptosis, and activates and recruits immune cells and sensitizes to immune therapy. And we published this back-to-back with Daniel de Carvalho's group. Beyond methylation, we and others have shown that when you add HDAC inhibitors, you can increase this response, and I'll tell you a little bit more about that. And others have nicely shown that when you inhibit histone methyltransferases, enzymes that add repressive histone marks, or enzymes that take away activating histone marks, you see the same response. Lastly, tumors that exhibit mutations in chromatin regulators have upregulation of TEs and interferon signaling baseline. And so I know this is a busy slide, but I wanted to summarize about 9 years of work and show that when you are treating cells with something that perturbs chromatin compaction, one of the biggest phenotypes is transcription of transposable elements where that wasn't occurring before, and activation of interferon signaling. And this can occur in a couple of ways. These are the kind of hypotheses for how this double-stranded RNA is being sensed. One is that you could have transcription from kind of both sides of these elements forming double-stranded RNA. One is that you can have similar TEs in different places coming together to form imperfectly paired double-stranded RNA. And the one that I think we have the most data for is transcription of inverted repeats that leads to RNA hairpins. And sensing by, there's a lot more evidence for MDA5 right now than RIG-I. So why does this matter? We were able to combine epigenetic therapy with anti-PD-1, immune checkpoint blockade, in an aggressive murine ovarian cancer model. And looking at survival, the black curve are the mice with no treatment, the gray is anti-PD-1 alone, and then the orange is the combination of epigenetic drugs with anti-PD-1. And so you can see a sensitization there. So because we saw this positive impact of the HDAC inhibitors and the DNMT inhibitors in our model, we wanted to understand more about how these epigenetic modifications together regulated the TEs and then the resulting downstream interferon response. And this was work led by my postdoctoral fellow, James McDonald. And here we're just looking at RNA sequencing after HDAC inhibitor, DNMT inhibitor, or the combination of both in 4 different ovarian cancer cell lines. And you can see a broad upregulation of these TEs, dominated in light blue here by the LTR ERV elements, just the same ones that were actually increased during cancer progression. And in general, we see that HDAC inhibitor alone, this is just looking at locus-specific level, we don't see much upregulation. It's driven by removing DNA methylation and a bit of an increase when you add the HDAC inhibitor as well. Unsurprisingly, because we're using a demethylating drug, we see a loss of methylation of these elements in the DNMT inhibitor treatment. And James also wanted to know how much what we were seeing in these cell lines overlap with what was happening in the clinic. And we were luckily able to access RNA sequencing data from a trial run by Daniela Mattei, where they used guadocytabine, a prodrug, a newer DNMT inhibitor, to sensitize ovarian cancer patients to carboplatin. And here we're looking at RNA sequencing data comparing post versus pre-biopsies, and every column is a patient here. So we have 8 different patients, and you can see similar upregulation of these transposable elements. So in patients, they're being upregulated as well by these drugs, again, dominated by these LTR-ERV elements. And when you look at the overlap between cell lines and patients, both at the family level and at the individual locus level for these TEs, it's actually much bigger than we expected. So here, it shows that what we're seeing in the cell lines mimics what's actually occurring in the clinic. Another question we had looking at our cell line data was the role of p53. And so again, in this disease, it's almost all p53 mutant. And when I discuss p53 mutations here, usually I'm talking about mutations in the DNA binding domain, sometimes called hotspot mutations, that are going to change p53 so that it no longer has its canonical cell cycle, DNA damage effect, but still can bind to different regions of the DNA and exert transcriptional activity. And we're interested in it because in model organisms, p53 represses TEs. John Abrams has shown really nicely that p53 represses line 1 in human cells, but Ting Wang has shown that p53 activates ERVs. And if you look at leaf-from-meni fibroblasts that are null for p53, these do upregulate TEs as well. So we used Nutlin, which was discussed extensively in the earlier talk, to stabilize p53 without causing DNA damage. We wanted to separate those two responses. So here we're looking at wild-type cell lines, and you can see very low level of p53, and it's induced by Nutlin, compared to these mutant cell lines that have that canonical overexpression of the mutant p53 like we see in the IHC from the beginning of the talk. On the right is just a positive control, showing that p21 target gene of p53 is only induced by Nutlin in the 2 wild-type cell lines, and not in these mutant cell lines or the SCOV3 null cell line. And James performed chromatin immunoprecipitation to see where p53 was binding upon Nutlin treatment. So here you can see that Nutlin caused p53 to bind to all these TE families and loci, again, dominated by LTRs. But we were surprised that the AZA or the DNMT inhibitor treatment also caused p53 to bind to these loci. And when we look at the RNA expression, we can see that Nutlin, p53 activation, increases TE expression, so does the DNMT inhibitor, but the combination of both increases this more than either one alone. So in this case, inhibiting methylation to open the chromatin plus activating p53 to increase its transcriptional activity had a bigger effect than either one alone. And this is all in the context of wild-type p53. Just as we were publishing this, Galina Slivanova had a great paper where she showed the same thing, where Nutlin activates p53 to go to chromatin, specifically ERVs here, causes transcription of ERVs, also represses LSD1 and DNMT1, and activates anti-tumor immunity, again, in the context of wild-type p53. Because most of our cancers are actually p53 mutant, we wanted to understand more what was happening in the hotspot mutants. So here is just Nutlin treatment in green and AZA in red in the wild-type cell lines, and you can see upregulation of 4 different TEs. This is another wild-type cell line where you see the same thing, both with Nutlin and with AZA. When you look at the null cell line for p53, Nutlin has no effect as expected, but the DNMT inhibitor also has no effect, which is really interesting. And then when you look at the hotspot mutant cell line, we see no effect with Nutlin, as expected from the previous talk, but we do still see an effect with the DNMT inhibitor treatment. So having some p53 protein there seems to work in this case. But because these cell lines have many differences besides their p53 status, we wanted to create a better controlled model and made an isogenic cell line with CRISPR, where we had a wild type and then an R175H mutant. So here you can see, again, with Nutlin treatment, the wild type, it induces p53, and the mutant is their baseline. And we do see a higher expression of TE's baseline in the mutant cell line. This is another volcano plot here, compared to the wild type cell line. So the R175H mutant p53 cell line has higher expression of TEs genome-wide. And in current work, we're doing cut and run to determine how mutant versus wild type p53 is binding to TEs across the genome. And so here, blue is wild type, red is mutant, and here is P21, where just the wild type should bind, and the mutant binds a lot less, compared to BRCA2, which is only a target of mutant P53, not wild type. And then we look at the TEs bound by P53, they're actually much higher in the mutant cell line. So we can add wild type P53 to our model here. And wild type P53 activates TEs. Mutant P53, I think we still need to understand what it's doing. It seems to activate them more at a baseline level. And we're doing experiments now to extend this to more cell lines and understand the binding partners of mutant P53. So next we wanted to understand about the editing post-transcriptional regulation of tRNA as well. And this gets us into the ADAR1 protein that was so nicely introduced in the previous talk. So we were curious about whether DNMT inhibitor treatment is going to increase this ADAR1 editing of the tRNA. And Daniel de Carvalho's group had already published that this tRNA is mostly what's binding to MDA5 here, mostly these hairpins from ALU elements, and that they are edited by ADAR1, which, because it's changing the RNA sequence, will change the binding of that hairpin, and thus it's sensing by MDA5. And this was work led by my PhD student, Stephanie Gomez, and she showed that AZA does increase RNA editing, but only in the P53 mutant cell line. So here we're looking at ALU and LTR elements, and you can see in the wild type background, the editing is not increased, but in the two R175H mutant cell lines, we do see an increase in editing. This is really interesting. We think this is likely due to the overall higher level of transcription of TEs in this mutant background. And Stephanie hypothesized that deleting ADAR1 would increase sensing of these hairpins after DNMT inhibitor treatment and thus type 1 interference signaling. So knock down ADAR1, and there's two isoforms of this protein, one that's baseline, one that's interferon inducible, and the knockdown takes out both of them. We do see an increase in interferon beta type 1 interferon secreted by these cells when you combine ADAR1, which is the hash mark, with DNMT inhibitors, which are going to be the maroon and the red colors. And we also see a reduction in proliferations. This is all cell intrinsic effects here, where you have interferon beta treated ADAR1 cells compared to GFP control, as well as DNMT inhibitor treated cells, which you can probably see better in the colony formation assays here, where the control compared to ADAR1 is azacitidine treatment and interferon beta. And we've shown this in multiple cell lines as well. So those are the cell intrinsic effects on cell proliferation. But we also see cell extrinsic effects, where combining the ADAR1 knockdown with DNMT inhibitor treatment increases secretion of chemokines like CCL2, CCL5, and CXCL10. And these two have been shown to be important in attracting lymphocytes to the tumor. So Stephanie took spleens from black 6 mice, isolated T cells, and activated them, also treated ovarian cancer cell lines that were syngeneic on black 6 mice with AZA, put them on the bottom of a trans wall, put the T cells on top, and then characterized the cells that came through by flow cytometry. And she showed that DNMT inhibitors did increase T cell migration in vitro. So here in the red and the purple bars, we do see an increase, but that the ADAR1 knockdown was not responsible for this. This was all a DNMT inhibitor effect. So then Stephanie combined these in a mouse model, where in gray, here's your survival curve of GFP with no treatment. The DNMT inhibitor in red increases its response. The ADAR1 in blue is actually much better than DNMT inhibitor alone, but the combination of both increased survival well beyond what we had seen before in this model, so it was much more powerful. And this was due to the reversal of immune suppressive microenvironment, where we see a reduction in M2-like macrophages, the ones that would be more tumor promoting, a reduction in suppressive myeloid cells, an increase in M1-like macrophages, and an increase in natural killer cells. And we also performed site-seq. So we're comparing the control to the ADAR1 knockdown. And you can see here, there's an increased infiltration of B cells and CD8 T cells. We were curious about what was driving this response. And so we inhibited type 1 interferon signaling in different immune cell types. So here is a survival curve. The solid purple is our DNMT inhibitor plus ADAR1 knockdown. The dashed purple is inhibition of interferon signaling. And you can see that this rescues almost the whole response. So it's all due to type 1 interferon signaling and its downstream effects. Inhibiting natural killer cells in blue had no effect. Inhibiting CD8 T cells in turquoise had a modest effect. And we do see more T cells in the spleens of these mice, so that there are increased T cells. And we also can see increase in specific lysis of tumor cells by T cells in these ADAR1 knockdown, as well as DNMT inhibitor treated mice. So we can add ADAR1 into our model here. And I think ongoing questions are really the role of B cells in this model, because we do see increased infiltration of B cells, and T cells do not explain our whole response that we saw. Okay, so in the last 2 minutes, I want to show you some data on how on what is occurring with TEs in ovarian cancer patients. And this is a really nice collaboration with Dale Garstead and David Botel, who did this study on short, moderate, and long-term survivors of ovarian cancer. And they've published this work showing that the long-term survivors of ovarian cancer have a lot of alterations in homologous recombination. Again, relevant to the previous talk of who is more likely to respond to chemotherapy. And also these long-term survivors had much more infiltration of T cells and B cells. So my PhD student, Kevin, first confirmed their published results where they showed that interferon gamma is much higher in these long-term survivors than short-term survivors. And this is a molecule secreted by NK cells and T cells to kill tumor cells. And then Kevin compared transposable element expression in long-term versus short-term survivors. And you can see an upregulation of TEs in long-term survivors. And 2 of them are shown here just as an example. And then he also looked at the locus-specific level in these long-term survivors. So again, we see a big upregulation of TEs. And here's one line-1 full length that's upregulated in long-term survivors. One that was actually downregulated called HERV-W up here is syncytin-1, and it has an immunosuppressive domain. Actually has a protein that's immunosuppressive, so that was really interesting. He also performed unsupervised clustering of these transposable element expression. And you can see that looking at the survival group up here in maroon, this really clusters with high expression of transposable elements. The long-term survivors seem to have higher expression of these LTR and line elements. It was even more clear with the locus-specific results, where you have long-term survivors clustering with high expression of TEs. Up here, we were looking at p53 mutation status, but unfortunately, all of the patients had some sort of p53 mutation, so we couldn't really make any conclusions comparing wild type to mutant. And Cedric Bichat had previously published that STAT1, which is downstream of interferon gamma, can actually induce some TEs, and this is a list of those TEs, but these TEs are not the ones that were induced in this model. So I was curious on what was causing this. Is it an effect of interferon gamma? It does not look like that, either at the family level or the locus-specific level. And so currently we're assessing methylation at the TEs that we have data for in these patients to determine whether methylation is a loss of methylation in these long-term survivors is causing TE expression to then activate immune signaling in these patients. So overall, I wanted to kind of summarize our work in that opening up the chromatin causes transcription of TEs that can be further edited by RNA editing. And then a whole part that I didn't talk about that is an area of much interest is whether peptides as well as full proteins from these can be recognized by T cells and B cells for novel anti-tumor immune therapy strategies. So I'd like to thank the people who did the work in my lab, as well as our funding and many collaborators. And thanks very much for your attention. Last talk of the day. I'm happy to take any questions.
Thanks for the talk. Any questions?
Okay, that was an excellent talk. Thank you. So I have a question about what you think is actually repressing the Ts in the context of mutant p53. Do you think there's another factor that is creating repressive chromatin at that space? And I'm thinking TRIN28-CAP1 mediated methylation of histones.
Yeah, great question. I think it's more that in the mutant p53 context, the ones that are upregulated have already lost their methylation in the context of cancer, right? And because we know p53 binds with different binding partners, we think that the mutant one is just binding with different binding partners, as has been shown for genes. The mutant p53 binds with different binding partners at genes, so we think the same thing is happening here, but we haven't done those experiments yet. Yeah.
Cool. Thank you.
Go ahead.
Thank you. Targeting TE is very promising. You just mentioned using epigenetic regulators like DNMTI and HDAC to modulate epigenetic reprogram. So besides TE signaling, are there any other signalings that are also perturbed by this DNMTI or HDAC? And how these signaling pathways are coordinated with TE-related signaling pathways?
Great question. So yeah, we know from the work of many other researchers that DNMT inhibitors and HDAC inhibitors can reactivate silenced tumor suppressor genes and thus cause anti-tumor effects. We try to distinguish our effects from that by inhibiting type 1 interferon signaling to show how much of our response is type 1 interferon dependent, and that's all from the TE.
Thank you.
Yeah, so my question builds a bit off, I guess, in line with Yadira's question. I found it really interesting when you did your sort of classic Weinberg transformation experiment that you get upregulation, which, some of those viral oncogenes will turn off P50. Is it just P50, loss of P53 or titrating away P53? Is that sufficient? And is that explaining why you see such broad upregulation across cancer? Or is there some other event going on to explain that?
Yeah, that's a great question. So SV40 is going to inactivate P53, right? That was what we thought at first. And so because that model, we only had that specific model sampled. In the FT cell lines, we actually have controlled cell lines, one that has wild type P53, one that's dull, and one that's mutant, and we're comparing those right now to determine exactly that question, because I think from what we know, I think you need more than just inactive or mutant or null P53. That seems to be one thing that you need, and you also need epigenetic loss of some epigenetic control.
All right. Thanks so much for the excellent talk, and let's thank all our speakers for this afternoon.